Hey everyone, and welcome back. Ready for another deep dive. Today we're shrinking down, way down, to explore the fascinating world of bacteria, specifically those that have a taste for something we all know and love: starch.
And trust me, the way these microbes have evolved to utilize starch is way cooler than it sounds.
Definitely. We're talking about tiny organisms, but they have these incredibly efficient tools at their disposal. Enzymes, right? They're like microscopic machines that help bacteria break down big, complex molecules like starch into smaller, easier to digest pieces.
Exactly. It's all about breaking down those large molecules into smaller components that they can actually use.
So it's like they're unlocking the energy stored within the starch, kind of like their own little carbohydrate buffet.
Precisely. And the key to this whole process, the enzyme that steals the show when it comes to starch, is amylase.
Amylase. Okay, so amylase is our star player. But how do scientists actually figure out if a bacterium has this starch-eating superpower? How do we know who's invited to the buffet and who isn't?
Well, that's where the Starch Hydrolysis Test comes in. And the beauty of it is it's actually quite simple. We're talking elegant in its simplicity.
Oh, I love those kinds of tests.
Yeah.
Simple yet effective. So walk us through it. What's involved?
Okay, so picture this. We start with a petri dish. You know those round dishes you often see in labs, filled with a special kind of agar?
Agar, that's like the jelly-like stuff that bacteria grow on, right?
Exactly. But here's the thing. This agar has been enriched with starch. So basically, we're setting up a bacterial feast. We let our bacteria grow on this starchy playground, and then things get really interesting.
Okay, I'm intrigued. What happens next? Do we, put it under a microscope and observe?
We could, but we have a more dramatic approach. We're bringing in iodine.
Iodine, like the stuff we use for cuts and scrapes. What does that have to do with bacteria eating starch?
Great question. It all comes down to iodine's color-changing abilities. You see, when iodine comes into contact with starch, it creates this really distinctive blue-black color.
Okay, I'm picturing it a dark, almost inky color. So we've got our bacteria on this starch-enriched agar, and then we add the iodine.
Well, that's when we get to see whether our bacteria have been chowing down on the starch or not.
Because if they have.
If they've been feasting on that starch, the area around them will remain its usual color, no blue-black in sight. But if they haven't been eating the starch,
then we'll see that telltale blue-black color, where the iodine has reacted with the starch, right? So no color change means the starch is gone, which means our bacteria have been busy producing amylase to break it down.
You've got it. It's like they're releasing these tiny little mouths called exoenzymes outside their cell to pre-digest the starch before they even eat it.
Whoa, talk about taking eating out to a whole new level.
Yeah.
But seriously, that's amazing.
It really is remarkable how these microscopic organisms have evolved such sophisticated ways to access nutrients. And all of this just from a simple color change on a petri dish.
So simple, yet so insightful. It tells us whether our bacteria are equipped with the right tools, in this case, amylase, to thrive in their environment.
Exactly. And to really see this in action, let's shift our focus to a couple of bacterial superstars. They're both commonly encountered, but they have vastly different lifestyles and relationships with starch.
Okay, we've got our head in the game, ready for a bacterial showdown. Who are our contenders today?
In this corner, we have Bacillus subtilis, a bacterium commonly found in soil. And in the other corner, hailing from the human gut, we have Escherichia coli.
All right, so it's Bacillus subtilis, representing the soil, versus Escherichia coli, straight out of the human gut. Is it going to be good?
It is a classic showdown, really highlights the diversity we see in the bacterial world.
So let's run the test. We've got our contenders plated on their starch agar. We hit them with the iodine. What happens? Who makes a break for the buffet?
Well, with Bacillus subtilis, we would absolutely see some action.
Oh, yeah. Tell me more.
A clear halo, nice and distinct, forming around the Bacillus subtilis colony. It's a textbook positive result.
Meaning?
Meaning our Bacillus subtilis is producing amylase, it's breaking down that starch, having a proper feast.
So point for Bacillus subtilis, which, when you think about it, makes sense, right?
Absolutely. Out in the soil, Bacillus subtilis is surrounded by decomposing plant matter. And guess what's a major component of plants?
Starch. So it's like Bacillus subtilis evolved with the perfect tool for the job.
Exactly. Having that amylase gives Bacillus subtilis a major advantage in that environment.
Survival of the fittest, even on a microscopic level. Okay, so what about E. coli, our gut buddy, holding up okay in this starch-eating showdown?
Well, with E. coli, the results are a little different.
Different how?
We wouldn't see that clear halo forming around the E. coli colonies.
Oh, so no clear zone. Does that mean?
It means E. coli isn't producing amylase. It can't break down starch. If anything, the agar around those colonies would be turning that distinctive blue-black, indicating a negative result.
So E. coli came to the starch buffet and didn't take a single bite.
Pretty much. Now, that might seem like a disadvantage, but remember, bacteria have adapted to thrive in very specific environments.
Right. It's not like our gut is full of, like, potatoes and bread waiting to be digested, right?
Exactly. The human gut is a completely different environment compared to soil. E. coli has evolved to utilize other, more readily available nutrients in the gut.
So it's found its niche, even without a taste for starch. And it's a good reminder that there are many different strategies for survival, even in the microscopic world.
Precisely. This simple test with the color-changing iodine, it gives us this amazing window into those different survival strategies. It highlights just how diverse the microbial world is.
It is mind-blowing when you think about it. All this information packed into a simple color change on a Petri dish. But I have to ask, we've been talking about bacteria, enzymes, starch. Why does this even matter for us, the non-bacteria out there? What's the big takeaway here?
That is the million-dollar question, isn't it? And it's a good one. Because understanding which bacteria possess specific enzymes, like amylase, it actually has massive implications. In medicine, for instance.
Oh, really? How so? So we've got these bacteria, some munching on starch, others ignoring it completely, and we can see it happening right there in the lab. But how does that translate to, practical applications, especially in medicine?
Well, imagine this. You're a doctor, and you have a patient with a mysterious infection. You need to identify the bacteria responsible so you can choose the most effective treatment, right?
Makes sense. Different bacteria, different treatments.
Right.
But I'm guessing this is where our starch-eating or not-eating bacteria come into play.
Precisely. Knowing which bacteria can break down starch can be a valuable clue in identifying them. It's like adding another piece to the puzzle, narrowing down the possibilities.
So it's kind of like a bacterial fingerprint at the scene of a biological crime. We see that clear halo. We know the culprit had a taste for starch.
That's a great way to put it. And it's not just about identification either. Understanding the unique enzymatic capabilities of different bacteria can help us develop more targeted treatments.
So those tiny halos on a Petri dish are actually giving us big clues about how to fight infections.
Oh,
Incredible.
wow.
But we've also talked about biotechnology, right? Where does this little enzyme amylase fit into that picture?
Oh, amylase is a busy bee in the biotech world. It's used in a wide range of industries, actually.
Like what? Give us the rundown. What can amylase do?
Well, for starters, it plays a key role in the food industry. Amylase is used to break down starches into sugars.
Okay, and that's important for...
Think brewing, baking, even producing sweeteners.
Hold on, are you telling me our sweet tooth is being satisfied with the help of microscopic bacteria?
It's true. Amylase, produced by those tiny microbes, is a key player in creating those sugary treats we all love.
That's wild, but it doesn't stop there, does it?
Not even close. Amylase is also used in the textile industry. It helps remove starch-based sizings from fabrics and the paper industry. They use it, too, to modify starch for better paper quality.
This is blowing my mind. From treating infections to making our food and clothes, these tiny organisms are having a huge impact on our lives.
It's a reminder that we share this planet with an incredible diversity of life, even at the microscopic level. And we've only just begun to scratch the surface of what we can learn from these organisms.
It's true. We started with a simple question. How do bacteria eat starch? And look where we ended up. Microbial metabolism, medicine, biotechnology.
Mm-hmm.
It's all connected.
It really highlights the power of scientific curiosity, doesn't it? It all starts with a question, a what if, and then, well, we put on our lab coats and explore.
So to everyone listening, the next time you encounter starch, whether you're baking bread, doing laundry, or even just thinking about it, take a moment to appreciate the unseen world of bacteria. They might be small, but they're mighty, shaping our world one enzymatic reaction at a time.
Couldn't have said it better myself.
This has been The Deep Dive. Until next time, keep those minds curious and never stop exploring.